Good morning, everyone. I'm Jim McCracken. It's my pleasure to welcome all of you to our June CART Distinguished Lectureship. And today's a special one of those in that this represents our annual RITFO Lectureship, which was established 4 years ago by Professor RITFO. And I'll give you a very brief introduction so as to not chew into our visiting speaker's time. But we're here, of course, to honor Professor RITFO. Dr. RITFO was a child psychiatrist trained in Boston and spent the entirety of his 30-plus academic career at UCLA. And Dr. RITFO, with several other colleagues here at UCLA, were at the vanguard of early autism research. Dr. RITFO himself was trained in the psychoanalytic tradition of the day, but he and others here at UCLA were focused on demonstrating that autism was not a product of failed mothering. And indeed, he, in his early, very provocative, often somewhat preliminary studies, nevertheless contributed to later studies that followed, demonstrating the high heritability of autism, the biological basis as shown by magnetic resonance imaging scans and EEG, sleep differences, and embarked on some very ambitious translational treatment studies as well. It's amazing to look back in many ways to see that several of those areas of work of Dr. RITFO, in a sense, seeded the field. And here, even at UCLA, we have a number of laboratories and investigators that are, in fact, in a sense, following up on Dr. RITFO's pioneering studies. Next, it's my pleasure to introduce today's speaker, who traveled great distance through much hardship to join us today. And it's great to be in person with Professor David Skuse from London. Dr. Skuse probably doesn't need much of an introduction to this audience, but he has been working the last 30 plus years in the area of autism and neurodevelopmental disorders. Professor Skuse trained at Manchester and then Oxford for general medicine before moving on to sort of the UK autism hub at the Institute of Psychiatry in London, where he worked with other autism research pioneers. David has taken on a very interesting career. In some respects, I liken it to Professor RITFO's in his willingness to take on somewhat controversial or provocative topics, maybe even doing a little disrupting along the way, which sometimes every field needs. But he's focused extensively on autism phenomenology, diagnostics, genetics, high functioning autism, and also sex and gender differences. And we're delighted that you could join us today, David, to talk about your work. So welcome.
Well, thank you very much. Well, thank you for inviting me. It's really a delight to be here. So what I thought I would do this morning was to talk about my involvement with this field, going back into ancient history, as Jim's suggested, and bring you up to date with what we're doing now. In fact, some of the analysis that I'm going to present, we did in the last 48 hours. And I hope I can be disruptive. I think I hope at the end you'll feel disrupted. And we'll see how things go. So I should start way back at the beginning, when I was an assistant professor, as you can see here, to So this eminent gentleman, Michael Rutter, later Sir Michael Rutter, who was one of the pioneers of autism research in the UK. This is a little pamphlet that I kept as a souvenir of my time with him, which I dug out the other day when I was remodeling my study. It was published in 1979. He talks about the chaos, the diagnostic chaos that existed. He was a somewhat irascible character, as Kathy will know. So that was sort of typical Mike word, really. There was chaos, and Mike was going to sort it out. How do we diagnose autism? And so this all happened while I was working with him at the Maudsley Hospital, as you can see there, and the Institute of Psychiatry is an academic institute attached to the hospital. So he influenced the first formal definitions of autism. Oops, sorry. And I think, you know, he questioned even back then, are there subtypes of autism? How should we classify children that show some but not all the features? I think questions that, you know, still demand our attention. And his diagnostic criteria were incorporated into the first formal account of autism in 1980 in DSM-3. But importantly, he also conducted the first genetic study of twin concordance. And I know Ritvo did also twin studies as well, but I think Mike's has precedence. And there was a letter to Nature, a very small letter. If you look up the original article, just a couple of paragraphs. But he said, in summary, we conclude that this systematic study of 21 same-sex twin pairs, in which at least one twin showed the syndrome of infantile autism, indicates the importance of a genetic factor in the etiology of autism. And as Jim's, you know, commented at the time, you know, there was an idea that perhaps this was caused by inappropriate mothering, you know, and primarily by environmental circumstances. So it's quite revolutionary. And then there was a further paper in the Journal of Child Psychology and Psychiatry, which is our country's leading child psychiatry journal, which described that study in greater detail, if anybody's interested. And he did this work with Susan Folstein, who was originally from the US. These were the early diagnostic criteria that they came up with. This was along with Eric Schoppler and Linda Lockyer, certain early onset, before 30 months, impaired social development, lack of social responsiveness, bizarre response to environmental stimuli, and the delayed and deviant language development. Now, those of you familiar with current diagnostic criteria may sort of recognize these are a little different. And of course, things did change. 1992, there was a revision to the triad of impairment: reciprocal social interaction skills, communication, nonverbal skills, lack of imagination, and repetitive stereotype behaviors, and inflexibility. Sure, some of you at least will remember that prior to DSM-5. But I'd like to point out that in 2005, using an instrument that I developed, which we called the 3DI, which was an interview. It was somewhat similar to the ADI, but it enabled us to break down individual symptoms of autism, to look at them in a more dimensional way than is possible with the ADIR. We found that actually that reciprocal social communication and use of language and other social communication skills correlated almost perfectly across the whole range, from typical individuals who are in the light blue there to individuals on the autism spectrum, which are in the upper part of the graph. Now, I think you don't need to be a statistician to see that's a straight line correlation. It's about 0.8. So we wrote this up and we sent it to a leading American psychiatry journal. And they said, but autism is a triad of impairments. obviously something wrong with your measurements. And the paper was rejected. And sat in the drawer, actually, for a very long time. I was somewhat irritated by that response. It was written with a couple of colleagues of mine, Joey Tang and Will Mandy. And Joey Tang later became the editor of a journal and she wrote to me a couple of years ago and she said, you got any papers for our new journal? We're launching this journal. And I said, oh yes, I've got the paper you wrote with us. Still there. So we rewrote that and it was eventually published in 2020. But of course, long after, DSM-5 had seen the error of its ways and redefined autism as bidimensional, as we suggested it should have been. There is an interesting paradox here, which maybe something Kathy can comment on later, but if you have the social communication component of autism, you might get the diagnosis of social communication disorder. I don't know how often that's been used, not very much. But if you have the repetitive and stereotype behaviors, what do you get? Nothing. Do they exist? Does anybody? And well, I'll come on to how we nearly managed to answer that question, but in fact failed in a moment. But anyway, so language delay is no longer a criterion, and onset before 30 months is not specified anymore. So there have been tremendous changes in the definition, and of course those tremendous changes have gone along with a terrific change in the prevalence, apparent prevalence of this condition. From 4 per 10,000, I would have failed my Royal College of Psychiatrists exam if I had not put 4 per 10,000 was the prevalence of autism way back in 1980. But now I believe in California, I was reading that maybe 1 in 25 boys has been diagnosed with autism. And in fact, there's a study coming out from the UK suggesting that in boys in early adolescence in the UK, the prevalence is somewhat similar. So yeah, huge changes. And about 20 years ago, I was at a meeting in New York and with John Constantino. Some of you will know John as the originator of the social responsiveness scale. And there were a panel of experts there. We were all discussing autism. And everybody but us said, well, autism is a very specific condition. And its prevalence is whatever it was then. It was about 5 per 10,000 at that point. And John and I said, I don't think so. And I remember saying to him, I think we're the dimensional men here, John, because he had done this study together with Richard Todd, sadly died shortly after this. But they showed that distribution of the scores on their autism scale, this social responsiveness scale, did not distinguish a subgroup of individuals, that they were continuous in the population. And in fact, we'd already done that with a very simple scale, which was actually the first autism screening instrument at the time. We called it a social and communication disorders checklist. And unfortunately, it just measured social communication skills of an autistic type. It didn't measure the repetitive and stereotype behaviors. But it was used in an epidemiological study in the UK called the Avon Longitudinal Study of Parents and Children. And that was based in my home city, as it coincidentally, of Bristol, city of about 600,000. And almost all the children, something like 97% of the children born, it's called children of the 90s. So these children were born 1990, 91. Almost all those children took part in this longitudinal study and are still being followed up. Probably the most comprehensive longitudinal study anywhere in the world. And when they were about 8 years of age, the person leading that, Jean Golding, said, David, do you have any measures of autistic traits? I said, well, no, there are no scales of autistic traits, but I'll make one up. And actually, coincidentally, I was working on a condition called Turner syndrome. I'm not going to go into all my Turner work now, but I was working on this condition where the girls were showing autistic-like traits. So the scale was actually based on Turner girls' behavior. It was very simple, just 12 questions and a total score of 24 possible. If you got a score of 0, 1, 2, if you got 2 in every question, you've got a score of 24. You can see the distributions there in that population. 8,000 children measured at age 8. And the lines there, the vertical lines, indicate the cutoff point between probable significant autistic traits and not. But it's a continuous distribution, very similar to what had been shown by John. And on the right, you can see that a further work by Elise Robinson, who's based here in the States. Elise looked at those traits over time, and we'll say some more about what she found in terms of genetic findings later on. But those traits were very stable. Basically, if you scored high at the age of 8, you would still be scoring high. Here it goes up to the age of 13, but in fact, they were followed up 16, 17 years. Those scores remained high. So they're sort of meaningful traits in the general population and stable traits in the general population, but define a lot more than 4 per 10,000 children. In fact, the prevalence rates actually not too different to what we're discovering in California now. I won't go into the details of that. But the problem was that now it looked as though autism was not rare and distinctive as Mike had believed. And indeed, a number of other people in the US as well have thought. If it lay at the extreme of a set of traits, what does that imply for its etiology? Now, the early genomic studies of autism were using linkage, using family members. In fact, they were looking for twin sibling pairs that shared autistic traits and trying to map regions that contain genes that predispose to autism. And that wasn't proving to be very successful. I've got 2 papers here from 1999. One from saying we expect to be able to narrow our search for autism susceptibility genes to a small number of genomic regions, those are the optimists. And then Neil Reisch and colleagues saying, much the same time, positional cloning of susceptibility loci by linkage analysis may be a formidable task, and formidable it proved to be, and was eventually abandoned. Now, around 2005, I visited Cold Spring Harbor. Some of you may know it's on Long Island. And the chancellor of Cold Spring Harbor was Jim Watson at the time, of Watson and Crick and DNA. Yeah. And Jim said, would you like to run a summer school? Sounded attractive idea. It's a beautiful spot. So, okay, I said I'd like to do one on the biology of social cognition. And we ran that for several years, actually, at Cold Spring Harbor. But in the time that I was there, I got to meet a guy called Jonathan Sabat and Mike Wigler, both very brilliant researchers, and of course, both very interested in autism research. I think Mike's still at Cold Spring Harbor, but Jonathan's now San Diego, I think, isn't he? And in 2005, there's this really neat article, I think, by Erica Check, which is summarizing a recent finding. She called it Patchwork People. And it was the discovery that we all have these copy number variants, which I guess everybody here knows what they are. But that was a big surprise, 2005, that our genome is full of these small deletions and duplications of chromosomal material. And on the back of that discovery, Jonathan started looking for whether these might be more common, or certain of them may be more common in people with autism. And this paper was published in Science in 2007, and I made a small contribution to that. And it's one of my most cited papers, I think, number 20 of the author, but nevertheless, it was a very exciting time. It was this new discovery a strong association between de novo, i.e. non-inherited, copy number mutations with autism. And around the same time, there was, in fact, increasing recognition that many relatively common inherited genetic variants or alleles contribute additively to the overall risk of severe mental disorders in general. So, for example, 2006, it's not that long ago, is it 17 years ago? Predictive testing for complex disease using multiple genes, fact or fiction. And of course, many people now thinking we might be able to use polygenic risk scores to predict who's at risk of all sorts of diseases, not just mental diseases. But there was a suggestion, that polygenic risk contributed to schizophrenia or bipolar disorder, and so on. Since then, whether polygenic risk carries significant specificity for major disorders, including autism, has been questioned. I think the jury's still out on that one. There's an interesting paper, Expanded View of Complex Traits from Polygenic to Omogenic. And I guess you guys will get the slides for this if you want to have a look. This was in Cell in 2017, discussing that issue. So shared molecular neuropathology across major psychiatric disorders parallels polygenic overlap. And then again, quite recently, actually, in 2013, there was a paper in the New England Journal which said clinical whole exome sequencing for the diagnosis of Mendelian disorders. So now this was taking the 3% or so of the genome that codes for proteins and looking in detail at the individual sort of nucleotide sequences and seeing whether those genes, some of those genes might be dysfunctional in people with neurodevelopmental disorders. And this paper, which was really the first paper of its type, 80% of children that they studied had what they call neurological phenotypes. And that included developmental delay, but it included autism as well, intellectual disabilities. And they came up with a 25% rate of molecular diagnosis. So this is something different to copy number variants, something different to polygenic variation. We're talking about de novo mutations, which are causing genes to become dysfunctional and putting you at risk of a neurodevelopmental condition. And it wasn't long before, of course, exome sequencing was applied to autism. So a couple of papers here which have been quite influential. This the one on the left with Mike Wigler as the senior author, contributions to de novo coding to autism spectrum disorder, and to recognize that both inherited and de novo genetic risk might impact on shared networks. So now moving from a time when we're just looking at some evidence of some genetic difference in people with autism to trying to understand how those differences might impact on neural networks that contribute to the symptoms of the condition. But way back in 2011, a group led by researchers in Cambridge had the idea of applying exome sequencing to children who looked as though they had some sort of genetic disorder. In the parlance of developmental pediatricians, they're called FLKs, or funny looking kids. They were very focused on the physical anomalies that may be associated with potential genetic disorders, funny looking ears and fingers and hair. Not so much, in fact, hardly at all, on the symptoms of the behavior or their autistic-like characteristics. I think this is a really important point. So I'm going to come back to this study in a moment, which underlies our own research. So DDD, as it was called, ascertained children who were thought likely to have some sort of genetic disorder on the basis of their physical phenotype. And about 90% of them had developmental delay. And arranged state-of-the-art exome sequencing at a leading genetic center in Cambridge, the Sanger Center, the Wellcome Sanger Institute. And this was a big study. I'll come back to it again. But they had quite a bit of success in discovering de novo mutations in that group of children. So essentially 3 classes of genomic risk for an autism diagnosis have been identified. This is a nice review by Jonathan Sibat and his colleagues, came out in Cell 2019, talking about gene mutations, talking about copy number variants, and we're talking about polygenic risk. So 3 classes of genetic anomaly. How important are those? Well, Thomas Bourgeron, who some of you may know, works in Paris and a leading researcher in this area, who Kathy and I have known for many years, ever since he was almost at school. Anyway, but he published this paper in 2016, suggesting that genetic risk was about 62%, environmental components unmeasured, about 38%, and also quantified the possible inherited or de novo CNV risks and the SV risks as well. I see that Dan Geschwin has recently commented that heritability could be as high as 80% on the basis of a study that was published in JAMA Psychiatry a few years ago. So I think genetic risk is very important. It lies somewhere perhaps between 50 and 80%. But it's probably towards the higher end of that. As I say, we don't really know anything about environmental risks at the moment, and this is something we're looking into. It I'll come back to. Polygenic risk is intriguing because it looks as though polygenic risk for traits in the general population is rather similar to polygenic risk for clinical ASD, as suggested by, this is another study from Elise Robinson, based on our work in the ALSPAC study. So this is using our measure, which when we developed it, we did do a twin study. We showed it was very, very highly heritable, high scores on our scale, which we call this social communication disorders checklist. And it turned out that SNP heritability, they collected DNA on these kids who took part in the ALSPAC study, SNP heritability measured for clinically diagnosed ASD, and that was in a couple of clinical populations in the States and in Denmark. 25% of those genetic influences also influence SC-DC scores. So there's some continuity between polygenic risk in the general population and polygenic risk in a clinically defined population. There's substantial and replicable etiological overlap. That's intriguing. And I think we're beginning, I should perhaps just say here that I think what we're beginning to see is a little possibly why there is this tremendous increase in apparent prevalence, because polygenic risk seems perhaps primarily to influence social communication traits, and kids are getting picked up because of their social communication problems. They're not getting picked up because of their sensory sensitivities or their rigidity, their cognitive rigidity insistence on sameness or their obsession with dinosaurs, or as Kathy's commented recently, lots of kids are interested in dinosaurs. It's when you can't communicate with your peers, and maybe it's something we could discuss later, but the demands, I think, on children's social competence is far greater now than it ever was in the past, particularly in the education system. So in the general population, this distribution of IQ in those with genetic risk is relatively normal. So polygenic risk doesn't seem to be associated with intellectual disability, or only to a very minor extent, according to this. These are data from Alsbach again. So what are those risk variants? Well, this paper, 2019, from Jacob Grove and colleagues, suggests a much greater role for spontaneous de novo mutations in those with ID or developmental delay. Severe ID, kids identified as having severe intellectual disabilities and autism, show far less heritability than those observed with normal or mild impairments of IQ. So the contribution of common variants, they concluded, may be more prominent in high-functioning autism, or what used to be called Asperger's syndrome. In contrast, the IQ distribution in clinically identified populations with de novo coding, so these are the exome sequenced samples, mutations, is well below average. And you can see here a couple of studies, one from 2014 from Associate Hall, I've mentioned before, and another more recent paper, which comes from Steve Shearer's lab, Trost et al. In Cell, November 2022. The dotted line is an IQ of 100, and as you can see, most of those individuals, particularly those with the SNVs, which is where that blue arrow is, have IQs well below that. So we've got a bit of an issue here in that we're talking about a set of genes that's influencing polygenic risk, but doesn't influence IQ. And we're talking about more specific genetic anomalies, leaving aside CMVs for the moment, in individual genes that, when they're mutated, will lead to autism, but almost always lead, apparently, to intellectual disability as well. This paper, Dan Geschwin's a co-author on this, and maybe Dan would like to comment on it, but came out quite recently, September last year, which suggested that compared ASD associated with polygenic risk and ASD associated with high impact de novo mutations, de novo variants were associated with lower IQ, but also with poorer adaptive skills, motor problems, but not core ASD features, apparently. And they question whether functional grouping of de novo variants could illuminate this observation. And I hope to illuminate this observation later, because that's what we've been doing. Another nice review, Jonathan Sabat's group again, also September 22 in Nature Genetics, concluding that from recent studies of polygenic risk in autism, the risk isn't strongly associated with learning difficulties, okay? We've seen that before. Common genetic variants that influence the risk of autism are different from those found to be mutated in de novo cases with intellectual disability. Common genetic variants convey a risk of other developmental disorders, such as schizophrenia and ADHD. So I think a pattern's beginning to emerge here, a number of studies coming to very similar conclusions. But that said, the phenotypic consequence of polygenic risk for ASD and de novo loss of function do overlap in some respects. And what this figure shows, if we just look at the red boxes, it suggests that on the left, down the bottom on underneath those circles, you'll see there's the de novo anomalies, loss of function missing, and on the right, the polygenic scores. And in terms of social anomalies, as measured, say, by the social communication questionnaire, there's a red arrow at the top, you'll see that both the de novos and the polygenic scores are associated with autistic-like characteristics as measured by the SCQ. Right at the top, interestingly, it looks as though polygenic scores don't influence repetitive and stereotype behaviors. So it was a pity we didn't measure those way back in the ALSTAT cohort when I had a chance to, a sample of 15,000 children. Potentially we could have come up with a scale to measure that in the whole population and looked at that in relation to genetic risk. But unfortunately, we didn't think about that at the time. And this was back in the 90s. This was a long time ago. So social behavior is influenced both by polygenic risk for ASD and de novo loss of function. But motor skills, interesting, that's the bottom of those two big blue circles, are only influenced by de novo variants. So, and now this paper from, this is Joe Buchsbaum's lab, Carl Satterstrom. And this, I was sent a preprint of this, and it was published literally a month before COVID struck us all and there was lockdown. So I've got two things rather quite linked in my mind with the shock of COVID, but the shock of seeing this paper, because what this paper claimed was that rare coding variants actually could be divided up. So I've been saying so far, all the evidence suggests that these rare de novo variants cause learning difficulties and associated autism, and you can't really pull the two things apart. But Carl Satterstrom and colleagues said, yes, you can. And I've done it. And there's a whole bunch of genes that I found are associated with, primarily with autism, another group of genes that are associated with autism, associated with intellectual disability. And furthermore, they said a couple of other things as well, which I thought were pretty revolutionary. I mean, one was that it depends when the genes are expressed. So prenatal gene expression would lead to ASD plus neurodevelopmental delay, postnatal to ASD predominant phenotypes, and also suggested that there was functional differences between these groups. So for example, regulatory gene anomalies, so those maybe chromatin anomalies that influence gene transcription in one group, and neuronal communication being more associated with ASD predominant phenotypes. So a lot of things in this paper which struck me as interesting and controversial and needed replication. Now, back again in September 22, this series of papers in Nature Genetics, they doubled down on this observation, and they made a further claim that some rare variants are associated with ASD, but not DD. So I will point out at this point, some of you may have noticed I've put the samples from which they're getting their cases, the logos there, I mean, SPARC, and I'm sure many of you will be familiar with these, the Autism Sequencing Consortium, Simon's collection. But they're tending to use the same samples. And one question I would ask, actually, is, does anybody know whether families sign up to multiple studies? Is there a way of checking that? I mean, it's just a question that's, okay, we can talk about that later. But nevertheless, these studies are all reworking essentially the same data sets. But anyway, nevertheless, they made the claim that it was possible, even though there was 86.6% of ASD genes are associated with developmental delay, that leaves 13.4% that aren't. So here are some outstanding research questions, and this one at the top was posed by Darren, looking a bit more friendly. How does specific genetic variants, specific mutations impact brain development in such a way the developmental outcome is autism? I mean, that's the big question. To which I would add, but how good is the evidence that some de novo variants impact social communication and other autistic traits independent of intellectual disability or developmental delay? Furthermore, the functional groups of de novo variants, for example, synaptic, gene expression regulation, cell adhesion molecules, have a differential impact on autism risk or specific components of the phenotype. How are we going to answer that? Well, I suggest that we could begin to answer that by turning this research on its head and taking a genetics first approach. Because all the studies I've talked about so far have started with a phenotype, start with 30,000 children with alleged autism, and then looked for genetic variants in that sample compared to a general population comparison sample that doesn't have ASD. There's not been a huge amount of work on genetics first studies. These are a couple of papers published relatively recently on autism in relation to genetics first. One of them was about CMVs, and I won't be saying any more about CMVs during this talk. I want to focus on de novo variants. And this was our unique starting point for a genetics first investigation. So this is the developmental disorders, deciphering developmental disorder study I mentioned earlier. And this article was published just a few weeks ago, actually, in the New England Journal. Genomic diagnosis of rare pediatric disease in the United Kingdom and Ireland. And there was an editorial. This editorial said, Writer improve the detection of additional variant types through systematic integration of methods designed to identify de novo mosaic, structural large insertion or deletion variants, as well as uniparental disomies, etc. They said their iterative analysis provides a framework for the clinical application of new discoveries and analysis methods in existing data. Now, we have access to this data. And Medical Research Council in the UK funded us back in 2015, which actually coincidentally the year in which DDD ended, data collection, funded us to do a national study of children with genetic anomalies that led to developmental delay. The red dots are where we collected the data from. You can see it was all over our country. We identified them through the local genetics clinics. Now, an important point is that DDD cases are anonymous. You can't go to a genetics clinic and say who took part in this study. You can say who was exome sequenced. So I think it's an important caveat that I say, I can't say for sure that every case that we identified with a de novo variant who'd been exome sequenced took part in DDD. But the NHS does not do exome sequencing. So they do microarrays, which is how the copy number variants were picked up, but not exome sequencing. Anyway, so you have a total of 3,400 families recruited into what we called Imagine ID. A case ascertainment was between 2015 and 2020, and we're now in the second phase, 2020-25, of following those children up. So this is a follow-up study that we're doing now of a group that was ascertained over a 5-year period. And of those 3,407 participants, 840 had disruptive SNVs identified by whole exome sequencing. So DDD, with its state-of-the-art analyses, said these children for sure have a genetic anomaly that's led to a phenotype. But they were not ascertaining for autism. They were ascertaining, as I say, for largely physical phenotypes, though noting in passing that 90% of the children had some degree of developmental delay. The demographic characteristics of that cohort, just the SNVs in our study, the 840, showed almost equal sex distribution. That's rather surprising. We know about inheritance in 80% of them. 65% we know with de novo, but 14.5% even of the de novos, of the SNVs were inherited. You can see the age distribution there between 4 and 19 years, largely biased towards the younger children. And the socioeconomic status of our samples reflected the UK socioeconomic distribution, as did actually their ethnic distribution. So this sample that we've identified is pretty representative of the UK population in terms of socioeconomic characteristics and ethnicity. The prevalence of autism in this sample was just over 30%. And in UK schools, as of 2021, the prevalence of autism was about 1.8%. So it's obviously a hugely increased risk in children who have an anomaly which was discovered by DDD. But the majority of them, obviously 70%, didn't have significant autistic traits. Our phenotyping was using the same methodology that was being used coincidentally by a group who were looking at children's mental health across the UK, sampling, I think it was about 10,000 children across the UK, and interviewing the parents, identifying the prevalence of mental health disorders in the general population, including autism. And we use the same techniques, exactly the same techniques, plus a few other things with our subsample of these 3,400 children. But what that means is we do have contemporaneous age sex match comparison data for the general population, collected exactly at the same time, 2017, same time we were collecting our data. And phenotyping included all children's mental health conditions. This wasn't a study just looking for autism. We got medical histories and adaptive functioning, but more recently, we've also got their school records through the Department for Education. And we've got their medical histories, which has taken a long time, 3 years, to get permission to get those, but we should have those very shortly. We've already looked at the school records, so I'm not going to present anything on that today. We have really comprehensive data on these kids. This comes from Kathy's paper recently in 2022 in the Lancet, the Lancet Commission on Research and Clinical Care in Autism. And this somewhat controversial idea that you could talk about, people who have profound needs, which in the paper, and Kathy suggested an IQ below 50. I've taken this is from the ABAS-3, but taken general composite score, adaptive composite score of 50 as a cutoff, just to show you, if you look at the kids that we've identified with de novo mutations, we find about 40% have ASD, an ID, and scores below 50, so have profound needs. There's about 35% of those with just intellectual disability without the autistic characteristics, somewhat similar. We've published a couple of papers on this project already. Jean Wollstonecroft, postdoc working with me since the inception of the study, was published last year. And then shortly before that, Sam Chawner, who is working in Cardiff, where they're more interested in CNVs identified in the study, looked at CNVs associated with neuropsychiatric risk. But I want to address these outstanding research questions in the last few minutes. How good is the evidence that some de novo variants impact social communication and other autistic traits independent of intellectual disability? And this is what Carl Satterstrom and his colleagues have claimed. So let's take two papers. I've mentioned both of these before, the Fu et al. Paper and the Trost et al. Paper, both published towards the latter part of last year. Trost, Steve Shearer's group, Fu with working with Joe Buxbaum. Now, to some extent, I think they were looking at similar samples. It's very difficult to figure out how much of an overlap there was, but I think there was a pretty substantial overlap. What you'll see here are the genes that we found in the imagined cohort of the genes that they said were autism risk genes. Okay, so 72, Fu et al. Said 72 autism genes. The Trost et al. Paper, 134. Now, of the Fu et al. Group, we found 35 in our cohort. Of the Trost et al. Group, we found 43. And so we've put those genes there. It doesn't matter what the specific genes are. What you can see, the red genes are ones that were unique to each study. So all the black ones overlap. The red ones were different in each study. But nevertheless, all those genes were found within our cohort, not an insubstantial proportion of the total that were identified by those two quite recently published investigations. And these are the proportions of the cohort with ASD diagnoses in the dark blue by risk genes. So we've now looked, we've got those 48 genes, 45 genes overlapping between both papers and Imagine ID. And interestingly, we find 15 weren't associated at all with autism in our cohort. So you take a developmental disability as your ascertainment, we find no association with autism. And in fact, that red line is 50%. There's precious few of them are actually associated with autism, more than 50%. Same genes, same genetic anomalies. I'm not going into all the detail there, but we have obviously exquisite detail on the nature of the anomaly. Fu et al. Went on to claim that there were 36 ASD-specific genes. Now, we found only 7 of those. Those are the pink vertical lines there. Really only one of them was associated more strongly than 50%. That was P10, to those of you who are gene spotters. But I mean, I suppose you could say 80% were not identified in our cohort. So maybe they are more ASD specific, because we didn't find them where we ascertained for developmental delay and then looked for autistic characteristics. But we also found 59 genes that were associated with at least a 50% risk in Imagine ID. And interestingly, 13 of those had no score in the Safari index of risk. So this is really turning things on its head. You know, we take a genetics first approach and we find a whole bunch of genes that are really strongly associated with ASD that are not even in Safari. Okay, we need to, we obviously need to do more work on that. And then the other outstanding question we had was, do functional groups of de novo variants, synaptic, gene expression, blah, 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 have a differential impact on ASD severity or any specific components of the phenotype? And that's something else we've looked at recently. And the answer, this is work done by Nadia Bednarczyk and Lucy Raymond. Lucy Raymond's professor of clinical genetics in Cambridge. Nadia is a very brilliant young pediatrician who's working with me. So they've categorized all the SNVs in the Imagine ID cohort into 13 functional groups, which is rather more, by the way, than anybody else has done that I'm aware of, certainly more than was published in the Satterstrom papers. And those dark blue lines indicate the proportion that had an autism diagnosis. As you can see, each of the functional categories has an autism diagnosis, though the proportion of genes, let's say, associated with chromatin regulation is much higher than the proportion that are involved in ciliopathies, at the other extreme of this list. We can look at that another way. So this is the proportion of participants with an autism diagnosis by functional group. And there you can see, I think, rather more clearly that the dotted line across the middle is the proportion overall that had autism, was about 30%. And yeah, there is some evidence perhaps that genes involved with RNA processing have more prominence than those involved in cellular processes. These two extreme lines in the middle, one way above the dotted line, one way below it. Unfortunately, even with that pretty large sample, we don't have the power really to say whether those are statistically significant differences. But it does look as though, it's not as though, for example, synaptic genes, which is the fourth one in from the left, that's not popping up. Hello, I'm here. Because so many studies have suggested that that's really the key, but not in this group anyway. So take-home messages. How good is the evidence that some de novo variants impact social communication and other autistic traits independent of intellectual disability? Well, claims for specificity don't hold up strongly when we study those genes in a population. Ascertained developmental delays or intellectual disability was subsequently found to have autism. The genetics-first approach has, however, identified new genes, new risk genes that were not previously recognized. Do functional groups of de novo variants have a differential impact on autism severity or specific components of the phenotype? Now, I haven't presented those data in detail because I'm running out of time. And again, we've really only very recently been doing that analysis. But we'd anticipated some specificity. Population risk, however, seems to be somewhat similar across all functional groups to date. So I don't know, is this our take-home message? David Ledbetter, some of you may know, claim, there are no autism-specific genes, just brain genes. Thank you.